Okay, great. Well, thank you so much for the very kind invitation today. So the title of my talk is Somatic Mosaicism Reveals Clonal Distributions of Neocortical Development. So as was mentioned, I am now at the University of Colorado at the Anschutz Medical Campus, but I used to be at the University of California, San Diego, where we did most of the work that I'm presenting today. So for today, I want to start by giving you a little bit of an introduction on the cellular context of mosaicism and talk about the prior developmental insights from mosaicism that we have obtained. And then I will define the research questions for this talk. And then I will talk about using mosaicism as a lineage mark, specifically in the neocortex. I will talk about the approach and technology, and I will spend a little bit of time also on the bioinformatic approaches, given that this is a data science seminar. And I will then specifically talk about a finding on the distribution of mosaic variants across the neocortex and the left-right asymmetry in development. Before I go into the introduction, I just want to acknowledge the people whose work I'm presenting, because I am very honored to be presenting on behalf of our group. So this was really a great collaboration between 4 individuals who are all contributing shared first authors. So it was myself, Dr. Xiaoshu Yang, Dr. Johannes Schlachecki, and Dr. Danny Antaki. So Dr. Antaki and Dr. Yang mainly did the bioinformatic analysis and really the heavy lifting in the background, but I'm very confident that I can answer any questions you might have about our pipelines. Dr. Schlachecki is really our expert on cell identity and sorting of the neocortex from postmortem brain. And just as a plug, both of these gentlemen are on the job market this cycle. If anyone knows of any jobs that would be appropriate for experts on somatic mosaicism or brain identity and epigenetics, please reach out to them or reach out to me and I'm happy to forward it. And so all of this work was done in the lab of Joe Gleason at UC San Diego. So a lot of you probably heard introductions on genetic mosaicism because you have a lot of experts here on campus. But I just want to start by this idea that one of the very defining insights of the 20th century was that every cell in our body has the same complement of DNA. This is not necessarily obvious, but it really enabled technologies such as cloning or induced pluripotent stem cells. So however, as most of the rules in biology, it's not entirely true, because actually every single cell in our body accumulates genetic mutations or somatic mosaic mutations. And in some cases, these actually might change the phenotype of the cell. We are very aware of this in instances like cancer, where somatic mutations actually convey a selective advantage and cause a disorder. However, not all mutations are necessarily having an impact on phenotype, and actually they do accumulate in every cell throughout development and aging. So thinking about this from a lineage perspective, starting at the zygote here on the left, and having a stem cell and an intermediate stem cell, and then finally the postmitotic cell, we can think about mutations occurring throughout this developmental trajectory, both in development, in adult stem cells, but also in postmitotic aging cells. And we typically distinguish here clonal mosaicism and private mosaicism. So mosaic variants that might be shared among different sister cells because they occurred in a mitotic cell, or those that actually accumulate in aging cells. So for this talk, we mainly will focus on clonal mosaic mutations, and especially thinking about how we could actually use them to understand development. And another way to look at this is with this very standard ball lineage plot, where every cell gives rise to 2 daughter cells. In this example, for every division, one of the cells will inherit a mutation, a somatic mosaic mutation, and the more cells we have, the more mutations we accumulate. So when we then look at a ball of cells that would be coming from this one original cell, which is an oversimplification of development, as we all know, but it's kind of a useful toy example, we do see that these early mosaic mutations actually are now fixed in many cells, and you accumulate mutations across the lineage depending on where you are at this decision point. So when we look at what we would call allelic fraction, so how many mutant alleles you have over the wild type alleles. So for instance, a heterozygous variant would be 0.5 allelic fraction or 50% allelic fraction. We can now look at these mosaic variants, and you can see that an early mutation actually would be here at 0.25, but there's only one of these mutations because there were only 2 cells with this mutation rate. But then at the next step, we have more mutations at a lower allelic fraction and so on. In an ideal world, we would have this stepwise contribution of mosaic mutations at each division. But in reality, what we normally would observe is this kind of exponential decay that would reflect the exponential growth of these cells at a constant mutation rate. So when thinking about this now, not in the context of a ball of cells, because as we probably all appreciate, we are not just balls of cells, we actually have a lot of structured development happening throughout our development. So for example, we could imagine that due to a structure in the developing embryo, you will then have a subset of the cells that are chosen stochastically or by a more structured developmental process that will go into tissue such as the heart. But you also can have different subdivisions for the neocortex or the cerebellum. So the idea now is, because these two structures here are actually closely related during development, so they happen at the same spatial area, roughly compared to, for example, the heart, and also in terms of the germ layers, we now can see that there are actually shared mosaic mutations between these two structures. And this can really teach us something about development. So counterintuitively, what we can do conceptually is that we look at a fully developed organism and we test for these mosaic variants, and then we can reconstruct the development that happened much earlier during the embryonic development of this individual. So how do we typically detect mosaicism? And for these kind of approaches, we normally distinguish a top-down approach or a bottom-up approach. So this here was a great figure from the Brain Somatic Mosaicism Network, of which many people on campus, on your campus, and also like my old group were part of. And so here the idea was to conceptualize how we can go from a tissue sample to mosaic variants. So a very straightforward approach, at least conceptually, is to use bulk tissue and then perform deep whole genome sequencing, for instance, if you want to look at variants across the genome. And then you actually call mosaic variants in this deep sequencing data. So there are several advantages and disadvantages to this approach. A big advantage is that you're very comprehensive. So within your sensitivity that is determined by your read depth and the quality of your read, you actually can sample all mosaic variants at a certain level. The disadvantage is clearly that depending on your read depth, you're only sensitive down to a certain allelic fraction. So an opposite approach would be the bottom-up approach, where you actually go and start from an isolated neuron, or maybe a clonal cell if you can expand it in vitro. And then you perform whole genome sequencing through whole genome amplification or clonal expansion. And then you actually have the ability, at least theoretically, to find every single mutation that happened in the history of this one cell. So the advantage clearly is that you have potentially maximum sensitivity. The disadvantage is that you are prone to artifacts, but also that you're really dependent on how many cells you sample to get a more comprehensive picture. And depending on where you're sampling from, you might actually miss certain lineages. So what are examples of using these technologies? So a very beautiful one was from Lodato, Woodworth, Lee et al., which was actually from the Park and Walsh labs at Harvard Medical School. And so you can see here an example where they actually sequence individual neurons, determined the mosaic variants that are present in these neurons, and then secondarily genotyped for clonal mosaic variants to determine lineage trees. And you can see here an example of different variants that are spread across the entire neocortical half, or these two examples, or restricted more on the anterior side of the neocortex. So this has been a very interesting insight because it really reflects how we think about development currently, which is that we have first a patterning event from anterior to posterior, or rostral to caudal, and then we have the left-right split. And this actually was replicated by work from the Church lab, where they used engineered mosaicism, so mosaicism that was controllably induced and can then be retrieved from a mouse brain in this case. And you can see this map, as you would expect, that the four brain halves are more related to each other than the midbrain or the hindbrain. But this is something we have now seen both in human and in mouse on this larger scale. The other approach, which is the top-down approach, also has been implemented, for instance, by the Park and Walsh lab in a recent publication last year. So there, the idea was to start with deep whole genome sequencing across the different germ layers and organs of the different germ layers, and then do targeted sequencing of almost 100 bulk samples and single cells to really try to understand early embryonic developmental events and distribution of clonal mosaic mutations. And one thing that was easily appreciated from this beautiful approach was that there are these bottlenecks, as I described before, for example, for different organs. But here there are bottlenecks that are actually apparent already at the first division. So you can see an example of an individual that roughly equally contributes both lineages to the final embryo and the different germ layers. But here's an example of an embryo that is actually very asymmetric. So you can now appreciate that there might be bottlenecks, in this case, probably the trough ectoderm, which will remove cells from the detectable embryo that are appreciable through this clonal mosaic analysis. So what were the research questions that really motivated our approach? So the first question was, will repeated sampling have the potential to increase the developmental insights? So if we sample from more than one area, for like one of these tissue cubes as shown in the beginning, do we actually find more mutations, and can we actually appreciate mosaicism in more depth? We also were interested to see whether we can see these developmental bottlenecks within the neocortex, similar to what people have found for these very early developmental events. And finally, we also wanted to see what the contribution of these identified clonal lineages is to distinct cell types. So to do this, we actually started with the anatomical dissection and sampling of a 70-year-old female brain that we obtained through the UCSD pathology. And so one of the biggest differences for our approach was that because we directly interfaced with our pathology, we were able to obtain both hemispheres of the brain and the entire neocortex essentially for our experiment. Whereas if you go to brain banks, a lot of them have a paradigm where they would freeze half of the hemisphere and the other half will go to histology, which is very useful for a lot of approaches. But if you want to comprehensively sample across a neocortex, this is really a big limitation. So in our case, we divided the brain roughly in a prefrontal, frontal, parietal, occipital, and temporal area on both hemispheres. And then we took these neocortical areas and we biopsied extensively, ending up with a small central biopsy that we will use for our further analysis, and a large area after the biopsy, which you can see here. In addition, we took individual samples or biopsies from the cerebellum, the kidneys, the heart, and the liver as outgroup tissues. But in this study, we really focused on neocortex, and we did now extensive sampling like has been done by the Park and Walsh study. So we then homogenized these large samples to get an overview of the mosaicism within these really large areas of the neocortex, comparing them to a central biopsy that's in the middle and our outgroup tissues. For all of these, we performed 300x whole genome sequencing. So just as a reference, this is roughly 8 to 10 times deeper than what you would do for a normal whole genome experiment if you try to detect germline variants. We then performed mosaic variant calling, which I will describe in a little bit more detail on the next slide. And finally, we did a validation analysis phase, where for each variant that we detected, we actually performed amplicon sequencing in a massive parallel amplicon sequencing approach, or AMPAS, which used AmpliSeq for Illumina as a technology platform. We then confirmed variants and accurately quantified them across the 25 samples that we initially used to call these variants. We did it on peripheral lobar punches, which I will not be talking about today. We did it on fluorescence activated nuclei nuclei sorting sorted populations, and we also genotyped single nuclei. So given the audience, I want to spend one slide to talk about our mosaic variant calling pipeline that we employed. As I mentioned, we started with 300x whole genome sequencing data, and then we performed alignment to HG19, mainly for the reason that most of the methods that we are using are extensively tested for HG19, but not necessarily for HG38. And we performed indel realignment and BQSR based on the best practices that were defined by the Brain Somatic Mosaicism Network in a recent landmark paper. We then did a combination of different mosaic calling technologies to cast a very wide net. And this included Mosaic Hunter, which if you're interested, you have an expert on your campus, August Wang, who actually was one of the developers and took this really to the next level, also doing a single cell Mosaic Hunter version where you combine bulk and single cell sequencing. We performed MUTEC2 in a paired mode in STRALKA2, which is coming from the cancer calling field, where we compared each area that we sequenced to the heart to get a tissue-specific variance. And finally, we also used MUTEC2 in single mode, which allows you to detect a large variety of mosaic variants with very low specificity. And we employed Mosaic Forecast, which is a random forest classifier, to then prioritize mosaic variants. If you are familiar or heard about Mosaic Forecast, it's probably because it was developed by Peter Park and his lab and Jan Maidu. After this, we employed a set of heuristic filters that we have defined in the lab, which includes areas that are of low probability to have a real mosaic variant. And this then allowed us to obtain the tissue shared and tissue specific mosaic variant list. And then we did the quantification through MPAS as I described before. I also want to put a plug for a new method that my colleague Xiaoxu Yang developed in the lab. So he actually used our regular approach that we as humans use, where we look at the mosaic variant to try to understand whether or not it's actually real. And he developed Deep Mosaic, which is a control-independent mosaic variant caller using a deep convolutional neural network, so I could get it out. And so this is a method we are currently extensively testing and we want to integrate in our pipelines. And if you're interested in this kind of technology, feel free to look at the preprint and also reach out to him. I'm sure he would be very happy to talk about implementing for other people. I also want to mention that in addition to our index case, we also did anatomical dissection and sampling of 3 additional individuals from a validation cohort, which we obtained from the Lieber Brain Institute. And so we're very grateful for this collaboration. This really allowed us to test some of our hypotheses and confirm some of our findings. We did a much smaller approach where we only looked at 5 small biopsies, performed variant calling, and did the MPAS. In some instances, I will actually mention this validation cohort, especially in one instance where it did not validate what we found in our index case. So going back to this first individual where we did the most extensive sampling, we wanted to first take a look at what kind of variants we actually detected. So you can see here the number of somatic variants, and in total we were able to detect 259 mosaic variants that were confirmed with our MPAS approach, 69 of which were found in brain and organs. So they seem to be early developmentally derived and are really spread across the biopsies and across the different organs. Not surprising, given our very extensive sampling approach, the vast majority of our variants are found in the brain, and more specifically in the neocortex. Quite to our surprise, we found a large number of lateralized variants that were spread across the hemisphere, and we found a significant portion of variants, almost half of the total number, that were found only in a single sample in the brain, and the vast majority, all but 4, were found in one of these small biopsies. When looking at the maximum mosaic fraction that we found for these variants, not quite surprising, we find that the brain and the organ mosaic variants are actually found at higher allelic fractions because they happen early in development, which is kind of consistent with our very simple ball model that if something happens earlier, it will spread wider, but also it will be there at higher allelic fractions. Consistently, those that were only found in a single sample show the lowest allelic fraction. So when we looked a little bit more at these single sample variants, we actually found to our surprise that they're very unequally distributed. So you can see here, again, the number of somatic variants that are private, so only found in one single biopsy. The small prefrontal, frontal, parietal, occipital, and temporal lobes, small biopsies. And you can see that the temporal really has a massive excess of mosaic variants. And this was not a result of the quality of the DNA or the quality of the tissue, because for the shared variants, we did not see the same issue of detection. So we came up with all kinds of very fantastic and really interesting biological reasons why development really would increase the mosaic variants in the temporal lobe, only to use our validation cohort and find that it's not as predictable as we would have thought it would be. So here you can see the 3 additional individuals, and you can immediately appreciate that there's a very high variance between how many mosaic variants you find in one of these small biopsies. And so while we still find an overrepresentation of temporal lobes, I do not believe that this would reach the level of significance that we would need to really say that there's an increase in the temporal lobes, especially because of the more limited sampling for our replication cohort. What it does show us, however, is that there's a very unpredictable asymmetry of these mosaic variants that I think it will still be of interest to explore further, but also gives us some idea of how the range of these mosaic variants could be. So next we wanted to take a more global look at all the mosaic variants, and we did some correlation analysis and hierarchical clustering. So here you can see every single clonal variant of the 259 that we detected correlating to every other variant. And we did Pearson's correlation. And so essentially this tells you how similar the pattern of distribution and abundance of a variant is across all the geographies. So immediately one thing that we found quite surprising is that contrary to what we would have expected initially, where you would expect that anterior to posterior is patterned first, actually we found much better clustering on the left and the right, within the left and the right hemispheres. So a large part of this is driven by the high number of private somatic variants. But still, even so, we do find that there is a natural hierarchical clustering of left versus right hemisphere. And this is something I will come back to a few more times throughout the talk, but I just want to mention it also from our rough bulk analysis that we did initially. So we then wanted to zoom in into some of the variants again, and we decided we need a way for us to represent the mosaic variants and their abundance across the tissues. And so I have this one slide that actually explains to you how we did this, because we used this in the lab to look at these samples, and we think it's a useful tool to conceptualize how mosaic variant is spread. So for this, we basically took the brain and then we projected the different areas into what we call a geoclone. So you can see here that it represents the large and the small area of the different neocortical areas, the brain, and also the additional tissues. And in the color, we will actually show the allelic fraction. So we can look at individual variants and we can find examples such as this one, which is a widely spread variant across the brain and the organs. We also find instances where we have things that are only found in brain and unequally distributed across the neocortex. More commonly, we find lateralized variants that are across the entire hemisphere or within certain area of the hemisphere, or the variants that are only found in one single brain sample. So one thing that immediately struck us was that while these distributions are clearly extremely different, if you would just look at individual biopsies, they actually have very similar and comparable allelic fractions. So when we looked at this in more detail, so where we have the number of positive samples across tissues, so 25 being present in all tissues versus 1 being present only in one single biopsy, while overall at the higher allelic fractions, we do find that the higher a variant is abundant, the more commonly spread it is across all the geographies. This roughly breaks down at around 6%, where we lose this nice correlation. So what do we think happens here? So what we believe happens is that we look at different variants, one of which would be at the early embryo stage. And then we have windows of these small neocortical biopsies that seem to be sourced for a smaller number of neocortical progenitors. And then this would in effect result in something that has a very different spread between across all the tissues or within one single biopsy, but has the same allelic fraction. So why do I highlight this? So I think the reason we highlight this is because if you, for example, look at a disease like focal cortical dysplasia, where we do have an expert on the call actually, it is kind of important if you think about doing a surgical resection, and if you have a certain allelic fraction, whether or not it would be spread more widely and might actually occur. And so we believe that actually just the allelic fraction by itself is not a very good predictor of this, but it's something we still have to explore in more detail. But we do believe this is a very interesting finding because we see this breakdown of correlation below a certain allelic fraction. So what I showed you so far was really all derived from our bulk data sets. So this means we have all the different cell types of the neocortex. So while I show you here a nice developmental system where we have everything coming from the developing neocortex itself, clearly we have other cell types that are not developmentally derived from the neocortical unlock. To address this, we decided to do fluorescence activated nuclei sorting. And so this was really done in collaboration with Johannes Schlachetsky and the Glass Lab, who are experts on retrieving these cells from the brains. So in our specific example, we actually did the nuclear isolation, and then we performed staining for NEUN-positive neurons, OLIG2 positive oligodendrocytes, LHX2 positive NEUN-negative astrocytes, and PO1 positive microglia. So 3 cell types that are derived from the neocortical unblockade in development, and 1 cell type that basically invades later on. I also want to mention that because of the abundance and the quality of the stains, we were able to easily identify neurons and oligodendrocytes. We were able to identify astrocytes for most of the regions, but microglia turned out to be only able to be sorted from 2 areas. So these data we then represent in this plot here, where we have the different cell types. Here the neurons, for example, with NEUN positivity, across the different areas of the brain, and across the left and the right hemisphere. And we also plot the allelic fraction in a square root transform way. So with this, we now are able to look specifically at the individual clones within the different cell types. So here, for instance, And in the background, you have a geoclone that gives you a presence or absence plot of the mosaic variant. We see a widely spread variant across the different neocortical cell types. We see another example here. We have a left-only variant that's only present in the left hemisphere. One that similarly is only present in one hemisphere, but more restricted. Another example of this. And finally, we also have the variants that we actually would like to include for subsequent analysis, which are only present in the microglia here, but not in the neocortically derived cells. So one thing you might immediately realize is that what I showed you is a lot of variants that are actually either skewed in one hemisphere over the other, or actually exclusively present in one hemisphere, which is again consistent with our bulk analysis, where left-right was really the main determinator of whether of correlation. So we wanted to make sure that this was not just us specifically seeing patterns that we like, but actually a more global phenomenon. And for this, we performed this analysis here, where we looked at the normalized delta, so the difference of a mosaic variant, whether it's on the left or the right, or it's anterior or posterior. And so we only did this for variants within the neocortical derived cell populations, so neurons, oligodendrocytes, and astrocytes. So again, somewhat surprising to us, what we found is that the polarization occurs first along the left-right axis. So you see a lot of variants that are essentially polarizing left-right, and then they can be distributed anteriorly or posteriorly. So this was quite interesting because this really, again, is different from our original model where we would think that anterior-posterior is patterned first. And in the neocortex, it appears that there is a different hierarchy that is secondary to this overall neocortical Anlage determination, which splits clones left to right and then anterior to posterior. So as you know, our brain has 3 dimensions, and there's an additional axis of ventral and dorsal. So what about this axis? So one complication with analyzing this axis is that actually during development, so this would be an embryonic cross-section of the human brain, we do have cells that locally migrate or radially migrate in the neocortex, and those that tangentially migrate into the same areas. So basically, we can distinguish dorsal and ventral cells by their identity, as the dorsal derived cells actually are excitatory neurons, astrocytes, and oligodendrocytes, while the inhibitory neurons and oligodendrocytes will migrate tangentially into the same area. So we distinguish those by our mu-n positive marker, OLIG2 positive marker, and the LHX2 positive mu-n negative marker. So can we actually infer these kind of biological principles of development in our data? So we believe yes. So first of all, I show you a correlation of neurons and oligodendrocytes, and you can see a very tight, nice correlation between these 2 cell types that are both derived from the dorsal and the ventral. So when we now compare it to the astrocytes, you can see that first of all, they still correlate quite well. So this probably suggests to us that the dorsal ventral split is secondary to the anterior posterior and secondary and basically after the left right as well. But also the correlation is not as tight. And this is consistent with these cells not originating exclusively from the same area, but astrocytes only dorsally and neurons only dorsally and ventrally. And we can do the control correlation of LHX2 and OLIG2, and it's similarly correlated. So an additional way to look at this is to specifically single out excitatory neurons and inhibitory neurons. So this is much more challenging in terms of a sorting strategy. And so we did a limited experiment where we actually used TBR1 for excitatory neurons and DLX1 for inhibitory neurons. So when we correlated these 2 subpopulations of excitatory and inhibitory neurons, we still find that there is decent correlation between the 2 populations. We do find examples where they diverge, but we do not find any examples where, for instance, you would have a ventral population across the neocortex or a dorsal population, but we always find mixes that kind of reflect likely bottlenecks. So finally, we also wanted to look at single nuclei, and we did this by single nuclei massive parallel amplicon sequencing, or SNMPAS, where we did again our homogenization and nuclear isolation, and then we specifically sorted out 48 UN positive cells and 47 UN negative cells. We did whole genome amplification and then MPAS. So this is less sophisticated than the bottom-up approaches that I showed you before, where you do an entire whole genome amplification and genotype and sequencing analysis, but it does allow you to place individual mosaic variants within one lineage by the means of having them within one single cell. So we did this analysis and we performed a ranking of the variants depending on how abundant they were across the individual cells. And you can see that some variants go with other variants, so they are within the same lineage. In essence, what we were able to do is to define major non-redundant lineages that roughly account to 0.4 or 80% of all the cells. And out of these, 3 we really defined as the major lineages that appear to happen very early in development. So again, mind you, we did this all from one small area of the left temporal lobe. So based on this idea, we would hypothesize that the 2 lineages 1 and 2 might have derived from the 4-cell stage, versus the 3rd lineage might have derived from the 8-cell stage with a slight overamplification, or alternatively, it might have also arisen at the 4-cell stage with a relative reduction. So we now have this hypothesis that these are early lineages, but so are they actually distributed across the brain equally as we would expect? So to do this, we actually collapsed these lineages and looked at their representation across different areas of the brain and also within the tissues. And indeed, all of these 3 clades that we defined here are represented across the neocortex in large and small areas and the organs. While there are some differences, which probably reflect the developmental bottlenecks, we do see that these early lineages actually are across the entire geography of the body. So what does this really tell us? So I always talked about us not being a ball of cells. I think we can all agree on this. And this is really reflected in having these bottlenecks and these different lineages, especially later on, where we can see really specific separation of, for example, the left-right hemispheres in the neocortex. But we also are not C. elegans. So we do have individual steps where actually we have a good mixture of all the different early lineages that will then contribute to the different organs. So let me come to the conclusions first by revisiting our research questions. So will the repeated sampling increase developmental insights? So we believe that yes, because we can resolve additional complexity and additional patterns by looking at different areas of the neocortex and also by repeated sampling of similar sized areas. We do believe that these developmental bottlenecks are really represented across the neocortex in these unexpected abundance or unexpected distributions. And what we found was specifically this interesting and intriguing left-right separation that appears to be upstream of an anterior-posterior diffusion or separation. And when we looked at the contribution of identified lineages to distinct cell types, we find that these early lineages are equally distributed across cell types and across geographies, but late lineages follow developmental rules that largely are deduced from mouse with this additional addition of the neocortical hierarchy of left, right, anterior, posterior, ventral dorsal. And so, as I said, these patterns have this potential to help us understand pathogenic mutations and understand this potential spread of mosaic mutations, even in the absence of any selection. Finally, I want to reiterate this hierarchy of cellular diffusion barriers that we think we observe in our data. So we would define a cellular diffusion barrier as something that either physically or due to diffusion restrictions inhibits the free spread of these clones that we identified, here indicated by these black dots. And so from our analysis, we really believe that there is a hierarchy of these CDBs that is first left-right within the neocortex, then anterior-posterior, and finally dorsal-ventral. So with this, I do want to come to my acknowledgement. Again, I want to thank Joe Gleason, in whose lab we performed most of this work, other than some analyses that we did in my lab. I want to thank my fantastic co-first authors, Shashu, Danny, and Johannes. Other members of the Gleason at the Glass Lab, especially Xin Chu and Edison Lana, who helped with a lot of these experiments. I want to also thank the UCSD Pathology Services, the Lieber Institute for Brain Development, the organ donors and the family, as well as my individual funding that I had throughout this project and our lab funding. And the hosting institutions, UC San Diego, Rady Children's Institute, collaborated on the sequencing, and also my current employer, the Anschutz Medical Campus. So I do want to end with a shameless plug that we are very interested in hiring excited scientists who do want to work on the interface of genomic mosaicism and development. And we have positions available, and I can tell you that the campus is very beautiful and it's very close to the mountains. So if you're interested in genomic mosaic research and are close to the mountains, you should feel free to reach out to me. And I'm very happy to take any questions you may have. Thank you for your attention.
Thank you so much, Martin. That was really great. And I'm going to turn things over to Courtney to moderate the questions.
Sure. Yep. Thank you. That was an excellent talk. Please, if anybody has questions, you can raise your hand and we'll unmute you to ask, or you can ask us in the chat and we can ask for you. I guess what people are thinking about that. I could start with this is actually a very general question I was thinking about. So you mentioned obviously how we can gain insights into pathogenic mutations and the like from looking at this. Do you have an idea or a thought about, how frequent it is that sort of genetically caused neurologic diseases are going to be caused by these brain-specific somatic mutations that we essentially can't get at with normal genomic sequencing methods?
Yeah, I mean, that is a great question. It was actually like a big question within the brain somatic mosaicism network. So this was really one of the driving interests of this whole group. So we know from some disorders like focal cortic dysplasias that are very clearly caused by mosaic variants. And so these are normally in the mTOR pathway. And so we do believe that there might be some selection as well as some overamplification, but this is not entirely clear. So that's one class of diseases where we know mosaic variants are definitely playing a role, or hemimegalencephaly, where the entire half of the neocortex has a malformation and causes epilepsy. For other neurological diseases, it's still not as clear. So there's been some great work, again, by the Walsh lab, but also by others, that looked at mosaicism in, for example, autism spectrum disorder. And if you look at global mosaicism across different organs and across different tissues, you definitely have maybe 5% or so that are detectable even from blood. So that leaves a potential for what about mosaic mutations that are only in the brain and potentially at high levels. And so we have not as good a handle of this at the moment. So there was a landmark study, again, by the Walsh lab that was looking at this in autism brains. And they did not necessarily find a lot of causative mutations, but they did find some enrichment in certain areas that might be relevant for brain development or function. So I think this is still something that people are working actively on, and we have not really covered all of it, but there definitely will be a subset of each of these disorders that are caused by de novo mutations that may actually be caused by mosaic variants that might cause less severe versions of a disorder or actually introduce new phenotypes because of the asymmetry that is introduced into the brain. At least that's what I think will be what we find moving forward.
Yeah, that's really interesting. Oh, that's great. Let's see. We do have a question from Anna.
Yeah.
Yeah. Should be able to.
Hello. So when you were talking about the unexpectedly high temporal mutations and then not having that replicated in your replication cohort, was there any difference in the ages of the people in the replication cohort? And then if there was, do you think that played into it at all with the different levels of the mutations that you found?
Yeah, I mean, that is a great question. So there's definitely one thing that we were worried about, which might be, did we get any hippocampal tissue because of the ongoing controversy, whether or not and to what level there is proliferation in the hippocampus. To answer your question directly, so we don't think that there's a significant age difference. So I think that the age range is roughly from 60s to 70s. So we don't think that should make this difference or underlie this difference. So what we do believe at the moment is really that this is just something that is stochastic across the brain. So we do have some areas that have higher mutation rates than others, and we're not quite sure whether this is specific to the area. As I showed, overall, if we look at the temporal versus prefrontal, which is where we have the most data from, there is definitely an enrichment in the temporal, but whether or not this is significant is hard to say at the moment, just because of the variation between individuals. But yeah, it's definitely something we thought about, whether age might have a role, but we don't think that our cohort is different enough between the replication and our initial index patients to explain this difference.
Interesting, thank you.
Sure, thank you.
Well, we'll see. A couple more questions. I actually had another question. So and actually the difference between people was one of them, but so so you mentioned, sometimes there can be an expansion or a kind of a decrease in the frequency of some of the mutations. So it's like higher or less than expected given which developmental time point it came in. Do you think that those are, and you mentioned bottlenecks being one big reason for something like that. Do you think it's mostly kind of random stuff like that, where just some cells or mutations get dropped out of the population, or could the mutations themselves be conferring some advantage or disadvantage? I guess from there, like the 259 variants you looked at, did any seem to impact functional regions or anything like that?
No, that's something that we definitely looked at. And so we could not find any reasonable annotation that would suggest that those 259 variants would cause any selective advantage or disadvantage. So, I mean, I guess we also wouldn't really expect to find something with a disadvantage just because you then wouldn't see it. So that's always the problem with looking at mosaic variants that might disappear. But so we don't think that any of the variants themselves, but we can definitely not exclude that, there might be another mutation and they are along for the ride and cause overproliferation. So this is something that does worry people who work on postmortem tissue, just because we can't really do appropriate experiments in humans, for various very good reasons. But yeah, so this is definitely something that we have been thinking about. We do believe, based on the patterns that we observe, that it is likely that these are truly neutral variants that are just along for the ride of development. And probably any differences we see might be inherent biological differences between, clones that have differential expansion, or for whatever reason, part of a clone dies, and therefore we see amplification of one clone over another. But at the moment, our default is that we believe it's normal development that is reflected in our data.
Nice, thank you. I think Pankaj has a question.
Yeah, no, that was a great talk, Martin. Thank you so much. I have a question, like, where do you see this field moving forward? Like, what's the next things that you want to reach to? Like, what do you see next 5-10 years where this is going to move towards?
Yeah, that's a good question, and a very important one, because I'm writing a lot of grants on this at the moment. But so one thing really is to understand these kind of developmental processes in more detail. So we, for instance, have a lot of interest in looking at some of these cell types more specifically to understand their development in more detail, or different areas of the brain. So that's kind of like where we are at. But I think a lot of people have a lot of interest in more disease pathogenic variants as well, just because the relevance for human health might be enormous. And I know there's like a very big initiative from the NIH that really tries to first provide an atlas of mosaic variation across many individuals, across many tissues. So actually the calls, I think, are currently, I think it just, the call just ended. But essentially they want to make a large database similar to GTEx to give everybody a good baseline. Because what we and others did was really doing it on a few individuals to try to understand very basic principles. And as you can see, even in our cohort, we could already appreciate the vast variation that we find across individuals. So I think this is like probably what is happening in the very near future, to just get a better understanding of how this process looks like. And clearly we won't be able to catalog individual variants like we do for EXAC or now NOMAD. But we will hopefully be able to understand patterns of what is like normal, what is abnormal, to understand different mechanisms.
Yeah,
and I think then, yeah, sure.
So now one more question I have is like, how much do you think this plays a role in like chronic disorders of aging, like Parkinson's or Alzheimer's or other ones?
Yeah, so there has been a wonderful study again by the Walsh Lab, who I mentioned a lot of times here. But so they did a very beautiful study on Alzheimer's. So where they show that there is actually mosaic variation that reflects biological mechanisms. So in their case, they found oxidative damage reflected in the genome in Alzheimer's. So definitely there is a way for using mosaicism for sure to understand biological processes as a readout or as a clonal marker. How much actually is driven by mosaic variation is still up to debate. There has been some work that looked at retrointegration, which has been kind of controversial, although very interesting, and also like, copy number variation that might actually underlie some of these different things, like copy number variation of APP, for instance, in Alzheimer's. So I think like for many of these disorders, there will be probably a portion for sure that is related to mosaic mutations that are driving. But we are not quite sure yet what this portion will be. And I think this is still something under active investigation by several groups around the globe.
Wonderful. Thank you so much, Martin. That was
Thank you.
great.
Thanks. We did have a question come in from Boxen Zhao. I think it's very related to what you were just talking about. So maybe you may or may not have some more comments about it, but essentially he comments that, most of the papers so far on somatic mosaicism are descriptive works, as he said, sort of cataloging what's out there. So when do you think the right timing would be for the field to move into really probing the functional studies, or has that already happened with some of these groups already looking, I guess,
Yeah.
these disease studies?
I mean, I think this is something that a lot of people are currently thinking about. There is definitely an interest from the organoid field to try to model some of these aspects. So clearly, because of the way organoids are set up, it's more likely to work really well for developmental questions. So for example, FCD has been done in a few organoid models where people try to understand more functional mechanisms. So this is definitely happening. So for other diseases, I think it is a good time to do these kind of studies. The problem is just, I think it's technologically very challenging because we already struggle to have really good, for instance, mouse models of autism. So there is, there are some phenotypes that work really well, but a lot of them are subtle or it's not easy to probe for them. And then if you put into the equation something that might be even more subtle, like a mosaic variant that's only present in like 10% of the cells, I'm just wondering how easy it will be to really probe for these mosaic variants. So I think the organoid work has been very promising for diseases that really have an impact on development and where you can track individual clones doing things that are not expected and understand it. But it has been challenging, and I think it remains challenging. But it's definitely, something that we all think about. But I personally, we are not really there yet. But I think some groups have made a lot of progress thinking about how you can integrate organoid models or other models to try to understand these mosaic effects.
Yeah, that organoid models is a very interesting point. And actually, that just reminded me of something that struck me, is the idea of the inducing the mutations in the mouse model from that paper a few years ago. Like that, it's very interesting way of kind of a forward way of looking at this as well. Okay, I think if anybody has any other questions, last chance. I think that we've covered all of them so far. So thank you again. That was excellent talk. Very interesting. And thank you to everyone coming. And it was a great discussion at the end. So yeah,
Thank you very much for the invitation. It was very nice to give a talk to your group. And if you have any further questions, feel free to reach out to me.
Great. Okay, thanks. Okay. I guess thanks everybody, and have a great rest of your day.